Okay students, now we will discuss the review exercise of chapter number 3 biotechnology, and it is on page number 42, and the start of this exercise is again with the MCQs. And the first MCQ is double helical strand of DNA are joined through. The first option is ionic bonding, covalent bonding, hydrogen bonding or metallic bonding, and the right answer is option number 3, that is hydrogen bonding. MCQ number 2, repeating units in DNA molecules are histones, nucleotides, plastids or chromatids, and the right answer is option number 2, that is nucleotides. MCQ number 3, smaller rings of DNA in host bacterium are chloroplast, chromosomes, plasmid or chromatids, and the right answer is option number 3, that is plasmid. Then MCQ number 4, two DNA molecules are joined by helicase, endonuclease, polymerase or ligase, and the right option is option 4, that is ligase. Now MCQ number 5, a recombinant plasmid is introduced in host bacterium cell through transfusion, transformation, transportation or transpiration, and the right answer is option number 2, that is transformation. MCQ number 6, golden rice is genetically engineered crop, transformed crop, tissue cultured crop or recombinant crop, and the right answer is option number 1, that is genetically engineered crop. Then MCQ number 7, choose the correct option about genetic engineering. Adding or removing the genes, study of extra chromosomal gene, study of gene, adding the plasmid vector, and the right answer is option number 1, adding or removing the genes. Now moving towards MCQ number 8, and that is the most commonly used biotechnological living tool in laboratories is bacteria, virus, algae or fungi, and the most commonly used is bacteria, option number 1. Number 9, synthetic plant seeds are produced through genetic engineering, transformation, tissue culturing or DNA fingerprinting, and the right option is option number 3, that is tissue culturing. Now MCQ number 10, microorganisms are used to remove pollutants from the environment in the form of bioprocessing, biocatalysis, bioremediation or biopollution, and the right answer is bioremediation. MCQ, the last MCQ number 11 is, as a result of DNA replication, the number of new formed strand is 2, 3, 4 or 5, and the right answer is option number 1, that is 2. Now we will move towards the B part of this review exercise, and that is answers these questions briefly. And the first question is biotechnology, that define biotechnology. What is biotechnology? Okay class, I have discussed this in your test exercise 2 as well, that biotechnology is a technology that utilizes biological systems, living organisms or their parts to develop or create different products for the benefit of human race. For example, the microorganisms are used in making bread, yogurt, cheese, vinegar and several medicines. Similarly, the processes like fermentation, tissue culture and genetic engineering, they are used for making many industrial products. Then question number 2 is, DNA strand is complementary, justify the statement. Now the meaning of word complementary is to combine in such a way to enhance the quality of each other, that two things combine in such a way that they will enhance the quality of one another or each other. Okay class, all of you knows that each strand of DNA is a collection of nucleotides, that is nitrogenous bases, sugar and phosphate groups, and there are 4 nitrogenous bases: adenine nucleotide, thymine nucleotide, cytosine nucleotide and guanine nucleotide. Now what happens, the DNA strand are complementary because nitrogenous bases present on one strand always make double bond with its complementary base. For example, such as adenine bonds with thymine and cytosine bonds with guanine. That is the reason each DNA strand is in a molecule is complementary to the other. Now moving towards question number 3, that what is the function of plasmid in bacterium? What is plasmid? The plasmid are the additional circular pieces of DNA and they are present in the cytoplasm. Now what they do, the plasmids have many different functions. They may have genes that can enhance the survival of the bacterium, survival chances that bacteria can live for long. Now how they increase the survival chances of the bacteria? Either by killing other organisms or by defending the host is bacteria, by producing the toxins. Some plasmids facilitate means make it easy, the process of respiration in bacteria. So some plasmids also make the bacteria easy in the process of respiration. Now question number 4 is differentiate between genetic engineering and tissue culturing. Genetic engineering enables the genes to get transferred from the DNA of an organism into the DNA of other organism. Now note this, DNA of one organism into the DNA of the other organism. Here both are alive and real. But in tissue culturing, what happens? In tissue culture is a technique of reproduction which involves the transfer of tissues. The genetic engineering involves the transfer of DNA and it involves the transfer of tissues or cells from an organism into a suitable culture medium to produce a whole new organism that is identical to the existing organism. Now in genetic or engineering, both are alive, one organism to other organism, but in tissue culture from the organisms to the artificial environment. In genetic engineering, DNA is transferred, but in tissue culturing, the tissues or cells are transferred. Okay, next question number 5 is how does biotechnology serve in the healthcare? Okay, several genetically engineered medicines like insulin, polio, vaccines, penicillin, etc. are available in the market. Okay, class, previously these products were not easily available in the market. Let's take the example of insulin. In past, it was extracted from the pancreas of animals. From where we get insulin? From the pancreas of animals. But nowadays, it is produced using the biotechnology techniques and is easily available in the market. Similarly, different antibodies, anti-allergics that will fight against the different allergies like dust allergies, pollen allergies and seasonal allergies are also biotechnological products that are commercially available. Due to these biotechnological products, years have been added in life because we get proper treatment on time, proper medication on time, and this is due to biotechnology. Okay, now question number 6 is environmental bioremediation is an application of biotechnology. Explain the statement. Okay, what is bioremediation? Bioremediation is the field of science that deals with the removal of pollutants from the environment using bioagents such as bacteria, algae and some fungi. Now what they do, these bioagents are used to break down the pollutants into harmless natural compounds, like they can degrade the dead animals and plants and bring valuable nutrients back to the earth. The biotechnological techniques use these microorganisms in a way to remove the pollutants from the environment. Now question number 7. Why DNA is needed to replicate? All of you know that when cell division occurs, the parent cell divides into 2 daughter cells. These 2 daughter cells further divide into 4 daughter cells and the process will go on. Now the new cells which are formed, they need all the complete components and set of informations as their parent cells have for a complete and proper survival. So the cell need to replicate their genetic material, mainly the DNA, when they tend to undergo cell division. This process ensures the retention of number of chromosome in daughter cells, because two daughter cells are formed, so the number of chromosome must have to replicate itself, so that equal number of chromosomes should be divided to the two daughter cells. Okay, the equal number, the daughter cells need the equal number of chromosomes as in their parent cell, and it can only be possible when the parent cell will replicate its DNA and make the copy of its DNA or chromosomes. Then question number 8 is, what would happen if DNA would not replicate before mitosis? When DNA replicate before mitosis, that phase is known as interphase. That phase is known as interphase. Now, if DNA does not replicate, what will happen? The complete set of information will not be passed to the daughter cells. They don't have the same number of chromosomes as their parent cells have, and they don't have complete instructions in the genes. If DNA does not replicate before mitosis, it will halve the size of genome that carries the instructions in the genes, and until each cell would die, which will probably not take long. Therefore, it is important that DNA doubles itself for the cell to split during mitosis. When they don't have complete instructions, when they don't have complete number of chromosomes, then they can't survive for long, and with the passage of time, which will not take long, the cell will die. Next question number 9 is, what does DNA replicates? When does DNA replicate? So DNA replicates before cell division, and that replication, that phase is known as interphase. Question number 10, how does vaccines work? Okay class, here how does the vaccines work? What do vaccines do? First of all, we will discuss the working of vaccines. That the vaccines, they contain weakened or killed pathogens. Now what are pathogens? They are disease causing germs. When a vaccine is injected into human body, what happens? The blood cells that are present in the body take the weak or dead pathogens as real ones and start preparing antibodies against them. These antibodies remains in the body. Okay, so when the real pathogen enter the body, the already present antibodies in the kill them immediately, and the body become protected from the disease. This is the way how vaccine works. So the answer will be, a vaccine carries weakened microbe disease causing, their toxins or their constituent proteins, which are called agents that encourages the immune system to identify the pathogen. Here, when the immune system and identify the pathogen, what will they do? They will start making antibody against that pathogen. When threat is identified means when the real pathogen attacks, the body starts developing antibodies against it, which reside in the body. And when a real pathogen enters into the body, these antibodies kill it all at once and protect the body. Now first, the dead pathogens were introduced into the body, and the body make antibodies against it. These antibodies are stored in the body, and where they will be used? When the real pathogen, when a live pathogen will attack, the already present antibodies will fight against these real pathogens and protect our body from the diseases. Now the last question of this part is, suppose the rice crop produced in your area is destroyed by insect every year. Being a biotechnologist, how would you save the crops? Okay ji, being a biotechnologist, I will produce insect resistant crop. Now what are insect resistant crops? The insect resistant crops are obtained by genetic engineering, and they generally produce compounds that are toxic to the insects that attempt to eat the resistant plants. And the second is, or I will make effective environment friendly insecticides. Insecticides are used to kill insects, environmental friendly, that will not harm the environment. Now coming towards the C part of review exercise, and these are the detailed questions. Answers these questions in detail. Now here I have taken the screenshots or the pictures of your book, and I have marked each and everything along with the page number. Okay, the page number are given, the topic number are given, and all the pictures from your book are given. The first question is, explain the process of DNA replication. Now the answer of this topic is on page number 32 and 33 of your book, and topic is 3.2. The topic is 3.2. You have to learn the complete topic 3.2 from page number 32 to page number 33. The next question is, describe the relationship between DNA, genes and chromosome. And the answer to this question is on page number 33, and the topic is 3.3, relationship between DNA, genes and chromosomes. And this answer is on page number 33. Next question is, explain how genes are introduced into bacterium. And answer of this question is on page number 34 and 35, and the topic is 3.5. You have to learn the complete topic along with the diagram on page number 35. Next question number 4 is, list some biotechnological products used in daily life, and the answer to this question is on page number 36, and the topic is 3.6. Question number 5, explain the genetic modification in different foods that can increase the amount of essential nutrients. Now this is on page number 37 and 38, and the topic is 3.7, and you have to learn only the underlined portion. You have to learn only the underlined portions. All the headings are mentioned, that which are the headings, and all the points are mentioned which you have to learn. You have to learn only the underlined parts. Next question, question number 6 is, list general application of biotechnology in various fields. Again, the answer is on page number 38, 39 and 40, and the topic is 3.8, and again you have to learn only the underlined portion. You are supposed to mark all these lines on your books, and after that you will learn only the underlined part. Then here the page number 40, and then the next question is, explain how biotechnology allows meeting the nutritional needs of growing population. It is on page number 40 and 41, and the topic is 3.9. The answer to this question, that is question number 7, is on page number 40 and 41. Question number 6 answer is on page number 40 and 41, and the topic is 3.9, and here I have marked the headings, and I have underlined some part at the very beginning of the topic, and then you have to learn the complete animal food production, plant food production, and at the end of this page you have to learn the complete. You have to learn complete plant food production and animal food production. Okay, now this is the end of our review exercise, and I hope all of you have got it, and if you have any question then we will discuss it during our live sessions. Okay class, thank you and Allah Hafiz.